So what we're going to do is explore the Ensembl Genome browser, and then we're going to move on and have a look at what we call the region in detail as well, which is a way to explore genomic locations in Ensembl. So the aims of this first webinar, firstly, is to introduce you to what Ensembl is. Maybe some of you have a good idea about Ensembl and the different types of data that you can access through the browser. But we want to explain to you all the different types of data that are available and how you can navigate around the browser to access those different data types. And finally, it's also going to be really important for us to show you where to go for help and documentation. This is an introductory course, so while we will cover a lot of the basics, there is a lot more information available online in the help and documentation that will allow you to continue learning about and continue using Ensembl in the future as well. So the Ensembl Genome browser was first conceived of in the late 1990s, just prior to the publication of the first draft of the human genome sequence. But actually, genome sequencing had been occurring for many years before that. So the first organism to have its genome sequenced was a bacteriophage back in the 70s, back in 1977. Fred Sanger and his team sequenced the genome of a bacteriophage. So that was 5 kilobases in size. So by today's sequencing standards, that was very small. But that technology laid the foundations for more and more complex organisms to have their genome sequenced until in the early 2000s, we had the publication of the first draft of the human genome sequence. So that's 3 gigabases in size. Even since then, we have the advent of next generation sequencing technologies, which has allowed us as a scientific community to sequence many more genomes much more efficiently at a much smaller cost. So what we're really coming to now is an age of data, of genomic data, where we have almost too much to process in the time that we have. So there's a number of global collaborative projects that are aiming to sequence lots of genomes, and one of Ensembl's jobs is to annotate those genomes and make them available to the scientific community through our browser and our databases. So some of these projects, for example, the 1000 Genomes project, that was a project that aimed to sequence lots of different individuals from different human populations, again producing lots of genomic data, identifying genomic variants across different populations around the world and their allele frequencies. But also the Vertebrate Genomes project is a good example of an initiative, a worldwide collaborative initiative to sequence all of the vertebrate genomes, all of the genomes of vertebrate species that are currently existing on Earth. So essentially what we now have is the accumulation of lots and lots of raw sequence data. So the first thing to mention, and that's very important to remember, is that Ensembl is not a sequencing project. So Ensembl doesn't sequence any genomes ourselves. We rely on research groups and collaborations to sequence the genomes of different species and then to deposit those genome sequences into publicly available archives. So that's what Ensembl, this is the sort of data that Ensembl might inherit. So this is just a string of A's, T's, G's, and C's. So this might be a section of a genome sequence. And this is what Ensembl would import from these publicly available archives. But obviously, as you can see, without any annotation or without any context or other information here, this is really quite meaningless. So that's what Ensembl as a genome browser tries to do. We take this genome sequence, we annotate it with genes, we annotate it with information about variation data between individuals within a specific species. We annotate features that regulate gene expression. We perform some comparative genomics, and we make all this data available through the web. So Ensembl, as we have already discussed, is a genome browser. So we take the genome sequences of different species and we annotate them with all this information that we're going to explore over the course of the webinar series. But Ensembl is not the only genome browser. So Ensembl is the project that is based in Europe, that is making access to genomic data possible through our interfaces. But there are other genome browsers that do similar things as well. So there's the NCBI Genome Data Viewer, there's the UCSC Genome Browser, and they both take, they again, similarly to Ensembl, they take this genomic data and then they make it available through their own interfaces with annotation as well. Now, these are examples of online genome browsers, but there are also offline genome browsers as well. So IGV and JBrowse are also genome browser interfaces that you can use to analyze genomic data as well. So I've already alluded to the 4 types of data that we generally have in Ensembl. So these are the main 4 types of data that we're going to think about over the course of the webinar series. So firstly, we take the genomic assemblies. So as I've already mentioned, these are genome sequences that have been deposited into publicly available archives by different research groups and collaborations, for example, around the world. So they will, then we take these deposited genomic assemblies and we annotate them with an automated gene annotation pipeline. So for every species, their genome is imported into Ensembl. We annotate them with our automated gene annotation pipeline that Astrid is going to talk about in the next webinar. The second type of data is the variation data. So this is where we take both small and large scale sequence variation data and we link that up with allele frequencies, phenotype associations, and lots of other predictive information as well about variants. The third type of data is the comparative genomics. So this is where we take the gene models and the genome sequences from one species to another, and we compare them. So we compute things like whole genome alignments to look at conserved regions, and we also produce gene trees to infer ortholog and paralog relationships as well. The fourth type of data is the annotation of regulatory features. So Ensembl uses raw data, such as ChIP-seq data, DNA methylation data, to predict the existence of features that regulate gene expression, such as promoters and enhancers. Now, the regulatory data at the moment is only available for human and mouse, so that's obviously not, that data is not going to be available for people that are studying species other than human and mouse, but it's hopefully you'll see in the regulatory webinar later on in the series that we are hoping to extend this data to other species as more data becomes available for us to use and analyze. So to draw these 4 branches of data together, Ensembl has a number of features. So as I said, we have our gene builds, which is the gene annotation for over 200 different vertebrate species now. And for those different species, we also have variation data for a number of species where that's available. Comparative genomics, we have comparisons across all the species in Ensembl, and also the regulatory data display for human and mouse as well. There's some other things that you can do with Ensembl that we're going to touch upon throughout the series as well. So firstly, you can upload your own data to Ensembl to view alongside all of the publicly available data. We also have a number of tools that are available for data analysis and processing as well. So the variant effect predictor, the VEP, is a good example of this, where we have a tool that will allow you to analyze your own variation data, which Michal will speak about next week. There's a number of ways that you can interact with this data as well. So all of the data that I've been explaining so far, this is all available through the web interface, but you can begin to export the data on a larger scale as well. So we've got the Biomart data export tool, which you might find useful for exporting medium-sized data sets if you have a set of data that you want to export from Ensembl. And then you can also access the data programmatically. So if you know Perl or any other programming language, so Python or R, for example, you can use those languages to access the data through the Perl and our REST APIs as well. The best thing about all of this is that it's completely open source. So all of the data we have is completely free for you to take and use in your own work. So as I said, Ensembl has access to over 200 different species. So we've got over 200 different genomes available through Ensembl. And one thing that you might notice when you go to Ensembl is that all of the species, most of the species, are vertebrate species. So we only hold data for species that have a notochord. So obviously, if you have a keen eye when you're looking at our species wheel here, you can see human, as well as the great apes. You'll also see the other placental mammals as well. Sorry, you see primates and the other placental mammals as well. And we try and include conservationally, environmentally, and evolutionarily important species as well. So you'll notice that we have mouse as an important model organism in biological research, as well as a number of other mouse strains that are also used in scientific research. But then we also have some marsupials and monotremes. So these are obviously the other branches of mammals that are important in studying mammalian evolution. And then we also have a number of other birds, reptiles, fish, and amphibians as well. What you might notice if you look at the very top of this species wheel, species tree, is that we have also 3 non-vertebrate species. So we have C. elegans, Drosophila melanogaster, the fruit fly, and also Saccharomyces cerevisiae as well. So these are 3 non-vertebrate species that we also include in Ensembl that are important model organisms as well for scientific research. So as I said, Ensembl is a project for vertebrate genomes. So all of the data that you can find for the vertebrates and these other 3 representative non-vertebrate species is all available through www.ensembl.org. So this is the main Ensembl genome browser for vertebrate data. But we also have a sister project called Ensembl Genomes. So if you're studying a non-vertebrate, so a bacteria, a fungi, a protist, a non-vertebrate metazoan, or a plant species, then you can access the same types of data, so the genome sequences, the gene annotation, variation data, comparative genomics, through our sister project called Ensembl Genomes. So that's www.ensemblegenomes.org. So we're going to mainly focus on Ensembl for our webinar series, but we will jump out to Ensembl Genomes at various stages just to show you how these different interfaces are mainly similar, but how there can be some differences as well for various data types. One of the other important things to remember when you're using Ensembl and Ensembl Genomes is that we work in a release cycle. So every 2 to 3 months, we release a new version of Ensembl. And this is important, obviously, to ensure that Ensembl incorporates the most up-to-date data and has the most up-to-date annotation and analyses. So Ensembl 98 was the previous version of Ensembl. That was released in September of 2019. And in the few months that followed that, we looked for new genome assemblies, and we updated some software, some underlying software updates. We reperformed some of our different analyses, so we updated our gene sets, we reperformed our comparative genomics, updated variation data and the regulation data, for example, and we packaged that data all up and we released it as Ensembl 99 in January of this year. So Ensembl 99 is the current version of Ensembl that contains all of the most up-to-date data, but we do hope to release Ensembl 100 in just actually a few weeks. So hopefully what you'll see if you're using Ensembl maybe in a few weeks' time is that we've released our new version, Ensembl 100. Now it's important to remember that we release new versions of Ensembl because it means that your data will be updated over time. So if you're used to looking at a single gene, for example, you might find that actually the data associated with that gene might change from one release to the next. So what we actually do is we make the archives of Ensembl available as well. So I'm going to show you this in the demo later on, but you can access the archives going back for up to 5 years. So you can look at the data as it would have appeared in Ensembl either a few months ago or even a few years ago. So you can see how the data has changed over time. So when you're using Ensembl, it's important to remember that what you're actually looking at in the Ensembl Genome browser is what we call a genome assembly. So a genome assembly is a representation of a genome. So I like to think of sort of the Earth and maps as being representations of the Earth is sort of a good analogy to describe what a genome assembly is. So you can think of the genome as being all of the DNA within the cell. So that's actually the Earth itself. So that's what you look at from space, for example, when you're looking at the Earth. But the genome assembly, which is what we actually have in Ensembl, is a representation of the genome. So you can see it's sort of genome assembly can be thought of as a map, and obviously maps can have various information. They might have errors, they might have gaps, especially as technology improves over time. The maps can become more filled in. There's the areas that are missing information might be then included, and any errors from previous maps will be corrected in new versions as well. So a genome assembly is basically a map of the genome, and that's also important to recognize that the maps have coordinate systems as well, just how genome assemblies have coordinate systems to help us navigate around the genome and know where we're talking about. So when a genome is being sequenced, the technology at the moment doesn't allow you to sequence a whole chromosome in one go. So sequencing is limited by the length of the reads that can be produced by that particular technology. So generally what has to happen is that the genome itself is fragmented into short reads. So we have these 150 to 200 base pair reads that are sequenced. Then these sequenced reads are matched up where they overlap. So you can see that's the second part of my of this diagram here. And then the final step is to infer the genome assembly from these matched up overlaps. So you can see here, for example, there's these two reads that agree at this position. There's a C. So in the genome assembly, we have a C. And the same here, we have another C that's actually agreed upon by these four separate reads, 1, 2, 3, 4. So we have a C at this position in the genome assembly. Now this cartoon obviously makes it seem like it's a very neat and organized process, but what you might find is that perhaps there are regions in the genome that do not have any reads, where we will obviously not know what that sequence is at that position. So that might lead to a gap in the genome assembly. And this process of matching up these overlaps can also be tricky for the alignment algorithms if there are highly repetitive regions. So if there's repetitive regions in the genome, it can be really difficult to find where these reads overlap, and that can also lead to some problems in inferring the genome sequence as well. So it's just important to remember that when you're looking at Ensembl and any other genome browsers, that what you're actually looking at is the genome assembly, which is basically the inferred sequence based on where these reads overlap. So when we're making a genome assembly, what we actually have to do is we have to the genome is being fragmented and sequenced, and then we have to put it back together, a bit like a jigsaw. So what you can see here is that the individual fragments have been cloned into what we call bacterial artificial chromosomes, BACs, and these come from different individuals. So this might be a BAC clone from one individual, and this might be a BAC clone from another individual. And then we match up where these clones overlap to form our tile path. So this set of red blocks here, this is called the tile path. And then the overlaps between these clones are trimmed to form our contigs. So you can see here, this light blue block is one contig that comes from this clone, and this second dark blue block is a second contig that comes from this clone here. So a run of contigs together with no gaps is called a scaffold. So this is a scaffold here, and this is another scaffold here. And then finally, we use genetic maps, things like fish data as well, to assemble the scaffolds onto the chromosome. So this is the final step, of assembling a genome, which is where we have all of these scaffolds placed on the chromosome in the correct orientation relative to one another. So this is an example of the human genome. So the human genome is as an example of the highest quality genome that's been sequenced that we have in Ensembl. And you can see, in fact, that the human genome assembly, this single chromosome here, will actually be made up of more than 4 contigs, but this is just an example here. But what you'll see is that the human genome assembly, for example, is a composite genome, which means that each of the different contigs actually comes from a library derived from a different individual. So what you'll see here, for example, is that this contig might come from one individual, whereas the next contig further down the genome might come from a completely different individual. So this is going to be very important in the variation module that Mihal's going to present when we're thinking about genetic variation and allele frequencies. Before we start the demo, the final thing I wanted to mention is that we have different versions of genome assemblies. So as the technology improves over time, a single species might have a new version of the genome assembly produced, which will have perhaps a higher quality, and we try to include those in Ensembl when they become available. So using human as an example, the most up-to-date version of the human genome assembly is called GRC-H38. So the GRC stands for the Genome Reference Consortium. So this is the group that's responsible for sequencing and making the human genome publicly available. This also sometimes called HG38. But this is the human genome assembly that's the most up-to-date and the highest quality human genome assembly that's currently available. And this is the data that you'll find in www.ensembl.org. Now, before GRC-H38 was made available, before that was produced, the human genome assembly that was being used at the time was called GRC-H37. That's also known as HG19. So this actually just got 2 different names. So when you're using different databases, you might come across these 2 names, but they're essentially talking about the same genome sequence for human. Now, many people still use the GRC-H37 genome assembly, so we do have a dedicated archive, grch37.ensembl.org, where you can browse and access data for the GRC-H37 version of the human genome assembly. It just is a slightly inferior quality, so there are some gaps, and there are some errors that have been corrected in GRC-H38. But as we said, many people still use the GRC-H37, so we still make that data available for people to use. So what I'd like to do in our live demo for this webinar is firstly to have a look at the Ensembl homepage, have a look at some of the data that we can explore from this page itself, and then we're going to have a look at a region of the human genome on chromosome 4, just to see how we can browse a genome and look at the different data tracks associated with that genome, with that region of the genome. So what I'd like to do is jump out of my presentation, and I'm just going to go to the Ensembl Genome browser. So we can just type www.ensembl.org into our internet browser. So this is the Ensembl homepage. So just while you spend a few minutes navigating there, for example, I just want to talk you through some of the different features on this page. So firstly, at the very top, we have this blue navigation bar. So this will appear at the top of every page in the Ensembl Genome browser. So you can use the links here to continue your search or to navigate to a different area of Ensembl that you might want to find. So the logo is actually a link back to the home page. Then we have some links to some other tools. So we have a link to our BLAST and our BLATS tool, the VEP that Mihal is going to speak about in the third webinar, as well as our suite of other tools that you can explore as well. We also have a link to the Biomart tool, which will be the focus of our last webinar. And then we also have information about downloading data and also some information through the help and documentation as well. So if you click on help and docs, you can see this is where we organize all of the help documentation. You can see that there's background about each of the 4 different data types in the annotation and prediction section over on the right-hand side here. But we also have, for example, a section of frequently asked questions, as well as a glossary of terms in case you're just a bit confused about the different terminology that we use in the Ensembl browser. There's lots of other pages that you can read about the background data as well, so you can explore this when you need to in your own time. To get back to the home page, I'm just going to click on the logo in the top left-hand corner. So that will bring me back to the home page. And the next thing to notice is the blue search bar underneath. So in this light blue blocks, we have our main search bar. So this is the portal into all of the data in Ensembl, really. So in this search bar, you can search for gene names, gene IDs, genomic coordinates, variant IDs, phenotypes, lots of different things you can search here. So we're going to use this search feature a lot through the course of the webinar series. Over on the right-hand side, you can see we have information about the release number. So you can see that we're currently in Ensembl release 99. If you use Ensembl in a few weeks' time, perhaps you'll notice that this release number has increased to Ensembl 100, and that will mean that we've got a new set of data that you can begin looking at and browsing as well. Further down the page, you can see we have information about the different species that we have in Ensembl. So the panel on the left-hand side, underneath the search bar, allows you to browse the different species available. So we're going to do that in a moment. But the final thing that I want to show you, if you scroll to the very bottom of the page, is the link in the bottom right-hand corner that says View in Archive Site. So when you go to www.ensembl.org, you'll always come to the current version of Ensembl. But if you click on this link in the bottom right-hand corner, you'll find a pop-up window that shows you all of the archives of Ensembl that you can browse and explore. So you can see, for example, at the very top, we have the dedicated archive for the GRCH37 human data, but going back in time as well, all of the previous releases for the past 5 years. So if you know that you were using Ensembl in December of 2017, for example, you can click on the link there, and that will take you to the Ensembl archive. So you can see here the URL has now changed. So we've got the dec2017.archive prefix at the beginning of our web URL. And you can see the color of the page has changed as well. So it's now this brown color. And that's just to show you that you're not in the current version of Ensembl. This is essentially just a freeze of the data from December of 2017. And you can use this website exactly as you would have used it at the time as well. Now, to navigate back to the current version of Ensembl, I can just take this prefix at the beginning of my URL, so this dec2017.archive, and I can just add the www back in. So if I just change the prefix to www and then press enter, it will bring me back to the main Ensembl homepage. What I'd like to do now is explore the different species that we have available for Ensembl. So in this box underneath the search menu, we can click on the link that says View Full List of all Ensembl species. So here we can see a table of all of the species in Ensembl. So it's quite a large table. You can explore that in your own time. But the filter over in the top right-hand corner allows us to explore, to search for a species of interest. So I'm going to just type Homo to begin searching for Homo sapiens. So you can see here, this is the entry for human in the table. We have information about the name of the assembly, so it's the GRCH38 Human Genome Assembly. And then we have the accession number as well. So this unique accession number refers to the Genome Assembly. And it means that when you're looking at data in different databases, in different resources online, for example, you can make sure that the accession number is consistent across all of your analysis so that you know that you're analyzing the same data when you're using different databases and resources online. So to find more information about human, you can click on human on the name on the table, and that will take you to a species-specific homepage. So what you can see now is a search bar at the very top. This allows us to search the human data. And then underneath, we have information about the gene annotation, the variation data, the comparative genomics, and the regulation data. So you can find examples of these different data types if you click on the links. But what I want to explore now is the box in the top left-hand corner about the genome assembly itself. So you can see that we're in the current version of Ensembl. So we have the GRCH38 genome assembly here. This is our data that we're going to be looking at. But underneath that, you can see that we also have a link for other assemblies. So you can see here other assemblies, and then we have a drop-down menu showing us the other assemblies that are available for human. So if we click on GRCH37 and then click Go, you can see that we've now navigated to a browser that's dedicated to the GRCH37 data. So if I begin searching now, this will be allowing us to search for the human, for the data associated with human GRCH37 assembly. For the purposes of the demonstration, I just want to go back to the GRCH38 assembly, so we can just ensure that GRCH38 is selected from this box again, and then click on Go. So the final thing in this section that I just want to show you is that if you click on More Information and Statistics from the Genome Assembly box in the top left-hand corner, you'll just find some background information about the Genome Assembly. So over on the left-hand side, you can find just a description of who produced the Genome Assembly and when, so the consortium perhaps that's responsible for sequencing the genome. And you can find information about the gene annotation process underneath as well. And then over on the right-hand side, you have information about the size of the genome, so the length of the genome. You also have information about the number of coding, non-coding, and non-coding genes that have been annotated across the whole genome. So this is just giving you some summary statistics of the whole human genome. The other thing that I wanted to mention before we move on to having a look at a region of the Human Genome Assembly is the data in the Ensembl Genomes pages. So if you're studying a non-vertebrate, you'll need to go to our separate website, www.ensemblgenomes.org. So you can open a new tab in your browser and type www.ensemblgenomes.org. So you can see the page is laid out in a very similar way. You have this blue navigation bar at the top to help you navigate, but the important difference to notice are these white links over in the top right-hand corner. So these allow you to navigate to the different pages of Ensembl Genomes that have information about these different branches of non-vertebrates. So if you're studying a plant, for example, a plant species, you can click on Plants, and that will take you to plants.ensemble.org. So that's the website where you can begin browsing and finding all of the data associated with the plant species. So it works in exactly the same way as Ensembl, but obviously it's just through a different portal. You can see the logo in the top left-hand corner. So this is Ensembl Plants. You have this small white arrow next to the logo. So if you click on the arrow, you can just see that you can toggle between these different pages. So if you also want to have a look at a fungi, for example, you can click on Ensembl Fungi, and now you can see the fungal species that are available. Again, the color coordination just tells you that now you're looking at the Ensembl Fungi pages. Okay, very good. So the final thing that I wanted to do in this webinar is to show you how to navigate a region of the human genome. So I'm going to use the tab that I was just working in, looking at the Human Genome Assembly, and I'm going to click on the logo to go back to the Ensembl homepage. So once I'm on the Ensembl homepage, I can use my search bar to search for my genomic region of interest. So to search, all I have to do is click on the drop-down menu to select my species of interest, and then I can enter my region, my genomic region, into the search bar. So my region is on chromosome 4. So I firstly type the chromosome number, then I need a colon, then I have my start coordinate, which is 122.868.000, and then I need a hyphen, a small dash, and then my end coordinate, which is 122.946.000. So this is the format that you need to adhere to when you're searching for a genomic region. So it's always chromosome number, colon, start, hyphen, end. So there's no other spaces or punctuation marks. So once you're happy, you can click go. And that will take you to the location tab. So just while the page loads, I just want to explain to you how these tabs are organized in Ensembl. So at the very top, you can see we're in the location tab. This is obviously the genomic region that we searched for. But as you click on different features within the browser, you're going to open up different types of tabs. So you're going to see this in the webinars further along in the series. But when you click on a gene, then you'll open a gene tab. When you click on a transcript, you'll have a transcript tab. So there's different types of tab for the different types of feature that we're going to explore. So at the moment, we're just looking at the location tab, and you're going to see the other types of tab as we go through the series as well. Underneath this tab, you can see a menu on the left-hand side. So you can see at the moment, we're looking at the region in detail. So this is the page that you can see in the main part of the browser. But you can also use this menu to explore some comparative genomics, for example. You can look at this region in other genome browsers like UCSC or NCBI, or you can navigate and look at the corresponding region in the human GRCH37 assembly as well. Underneath the menu, you can see these 5 blue buttons. Now these allow you to interact with and configure the data that's on the main part of the page. So I just want to walk through these buttons very quickly because they're quite important in helping you find the data that you're interested in seeing in the browser. So the blue configure this page button, we're going to use this in a moment, but this allows you to add data to the view. So it allows you to add more information to this view. At the moment, we just have the default data set that's been loaded on the page, but there's more data that we can add, and we're going to do that in a moment. The next blue button is called Custom Tracks. There's online tutorials of how you can do this, but this is the way where you can upload your own data set to view alongside all of the publicly available data in the Ensembl Genome browser. The next button is called Export Data. So I'm going to click on that now. So if you click on Export Data, what you'll find is a pop-up window that allows you to export the data from this region. So there's a number of options that you can select. The first is the output format. So by default, we'll be downloading the FASTA sequence, but you can choose other data types and other formats that you might want to download using the drop-down menu here. Obviously, if we're downloading sequence, we will choose FASTA, and we can just ensure that the coordinates are correct for what we want to download, and then we will click next to download. I won't download anything now, but that's how you can download the sequence from this genomic region if you just want the sequence perhaps to input into a primer design tool or some other analysis tool that you're using. Once you've downloaded any sequences and you're happy, you can get rid of the pop-up window just by clicking on the tick in the top right-hand corner. And then the final button on the left-hand side I wanted to mention to you is the Share This Page button. So if you click here, this will create a unique URL that you can copy and paste and share with any collaborators or colleagues that you might have. So this is important because if you share the URL at the very top in your web browser, all that your colleague will see when they follow the link is the default page. However, if you've manipulated the page, added some extra data tracks that are not there by default, you can create this unique link that will allow your collaborator to see exactly what you're seeing on your screen. So it's quite a good way to share and collaborate with people who are using Ensembl as well as you. Okay, so the final things that I want to quickly walk through before we finish the webinar is just to talk you through the data that we have in the main part of the page. So you can see the view is split into these 3 consecutive images. So each image is more detailed than the last. At the very top, we have the chromosomal overview. So you can see here we're looking at human chromosome 4. This is the P arm of the human chromosome 4, you can see, then the centromere, and then the Q arm. And interestingly, you can see that we have this red box. So this red box is our region of interest. This is the region that we searched for, and then we begin to focus on this region when we look at the images further down the page. Outside of this red box, you also have these other red highlighted regions and green highlighted regions as well. So these are called assembly exceptions, and it's what you can do is you can click on them and find more information about what these assembly exceptions are. So they might represent some corrections to the original genome assembly or some haplotypic regions as well. So the highlighted regions that you can see here, these are assembly exceptions, and you can find out more by clicking on them and following the links in the pop-up window. Further down the page, you can see the next image is what we call the 1 megabase overview. So you can see the scale bar at the top here. This is 1 megabase. And now you can see the data is organized into tracks. So the first track to look at and to orientate yourselves with is the contigs track. So this blue bar represents the genome assembly. So just as I explained in my presentation, this alternating light and dark blue blocks represents the double-stranded DNA. So this is the contigs track, and then we have data in the other tracks that is aligned to the genome assembly, the blue bar. So the track underneath is called genes. So you can see the genes are represented as these blocks, and then the color of the blocks indicates the biotype. So Astrid is going to talk to you about the biotype of genes and transcripts in the next module. But for now, you can just understand that each of these blocks represents a single gene. So you can see here, this is the SPAT-A5 gene. This is the NOD-T6 gene, this small gene here in gold in the center of our red box. And then this is the FGF2 gene, this gold one here. So again, you can see our red box. This is our region of interest. And we're beginning to become more focused on this region that we originally searched for. So this is our start coordinate, and this is our end coordinate. If I scroll to the very bottom of the page, you can see that this is the region in detail itself. So this is the most comprehensive view of this region that we searched for. And this essentially is our red box. So this is our start coordinate over on the left-hand side, and this is our end coordinate over on the right-hand side. Again, you can see the data is all arranged into these tracks. So you can see the contigs track is our track of interest that represents the genome assembly. So this is the double-stranded DNA. And then you have the data arranged into separate tracks, both above and below the contigs, the genome assembly. So you can see, for example, we have this genes track here. And now you can see that the genes, instead of being represented as blocks, they're now represented as the individual transcripts that are made up of blocks and lines. So these are these individual lines and blocks, these cartoons, these are the individual transcripts of that particular gene. So this is one transcript of the NOD-T6 gene, and this is another transcript of the NOD-T6 gene. Again, Astrid is going to talk to you about the colors and the representation of the genes and transcripts in more detail in the next webinar. At the very bottom, you can see again there's a legend that explains the color coordination about all of the different features. So what I'd like to do now is just show you how you can add some data to this view. So the tracks that we're seeing at the moment are what we see by default. But if we want to add more tracks, we can go to the blue bar, the blue button on the left-hand side of the page, configure this page. So we can click on this option. And this allows us to search for and add new data tracks. So this pop-up window obviously shows us all the tracks at the moment that are active. So by default, we're just looking at these active tracks. But the menu on the left-hand side allows us to explore and find all of the tracks that are available to us. Now, there are lots of tracks, so we won't have time to go through them all. Obviously, in the exercises, you can spend some time to familiarize yourself with the different tracks that might be interesting to you. But I just want to add a couple at the moment to show you how this might work. So if you click on variation from the menu on the left-hand side, you can see all of the tracks that are available that show variation data. So where a box has an icon, you can see here this nomad data. This shows you that this track is turned on by default. So this track is currently on, whereas all of these other tracks are currently turned off. So if you want to turn a track on, you can just click on the box, the empty box, and then in the pop-up window, you can choose the formatting. So I can choose the normal formatting to turn this track on. So the LOVD variants will now be on. If I'm not sure what a track is, I can click on the information icon, and that will just give me a small piece of information, a bit of background about the data that will be found in this track. If I want to search all of the tracks, I can use this option in the top left-hand corner. So if I want to search to find tracks showing me data from the UniProt database, information about protein sequences, I can type UniProt into the search bar and find the tracks that have data associated with UniProt. So I'm going to add this track at the top here. So protein alignments from the UniProt database for mammal. So I can click on the track again, select the normal format. So once I've added the tracks that I want to view, I can save and close the pop-up window by clicking on the tick in the top right-hand corner. So the page is now reloaded, and you can see that we have the new tracks. So you can see we have the protein alignments from UniProt. These are alignments to the forward strand, and we know that these are alignments to the forward strand because we have this colored bar on the left-hand side. So wherever you see a track with this blue colored bar to the left-hand side of the track, you know that that is data associated with the forward strand. And then when we scroll down, we can see that we also have a separate track for the UniProt alignments from the mammalian proteins. But this time we have this yellow-orange colored bar to the left-hand side, which tells us that this is data associated with the reverse strand. The final track we added was this LOVD variation data track. And in fact, you can see there's an error message here that says that there are no LOVD variants found in this region. So what I might like to do is turn this track off now, once I've realized that there is no data to see in this region. So I can click on the track name. So here I can read more information, change the formatting, but also just turn that track off if I want to. So I can click on the cross to turn the track off. I can also move the tracks relative to one another. So if I want to move this variation data track further up the page, I can hover my mouse over the colored block on the left-hand side and click and drag that track further up the page. So I can move the tracks relative to one another to begin looking at the different data in relation to one another. The final thing that you can do that I want to very quickly show you is that you can manipulate the view here and scroll around the genome and zoom in and out using these options in the top right-hand corner. So at the moment, I have this dotted box selected in the top right-hand corner of my view. So that means when I click and drag with my mouse in the view itself, and then let go of my mouse, I'm given an option to either mark the region, so to highlight that region, or to jump to the region. So if I click jump to region, that will reload the page around that new region of interest that I just defined with my box. If I click on the double-headed arrow instead, that just changes the functionality of my mouse. So now if I click and drag, I can just move up and down the genome to explore upstream and downstream. The other options in the top left-hand corner of the window allow me to configure this image, so to add tracks, upload my own data, share the image. I can even export the image, and I can reset the configuration as well. So if I click on reset configuration, I can go back to the default view and the default tracks that were showing when I first arrived at the page.
The final thing to remember is that this view is interactive. So if you want to find out any information about the features in any of the tracks, all you have to do is click on them. So if I want to find more information about this transcript of the FGF2 gene, all I'd have to do is click on the feature itself in the view, and that will open up a pop-up window that contains more information about that feature. So here you can see I can find the name of the gene, as well as the gene stable ID and the transcript stable ID, which are links to the other pages in Ensembl that will also contain further information.